Good morning, students. Let me teach about Agarose gel electrophoresis in today's class. Let us see the outline of today's presentation, which includes introduction, procedure, applications, and references. So let me start with a simple term, what is meant by Agarose gel electrophoresis. So this is a molecular technique by which isolation and separation of nucleic acid, that is DNA and RNA, can be separated based on their charges. So this is a very simple definition. So this technique was introduced by Tiselius in the year 1937, and the very first step which is required for this thing is you need a very large DNA which can be broken down into small fragments with the help of restriction endonuclease. So once this DNA is cut into fragments, the next step will be separating the DNA fragments based on their charges. So separation of DNA fragments is done under the influence of electric current. So that is why it's termed as electrophoresis, where electro in the sense electric current is supplied, and phoresis in the sense here we are going to provide some supporting medium or the matrix which is a porous network. So here in the case of this procedure, before stepping into the procedure, let me explain what the electric current we use here and how do we prepare the Agarose gel. Okay, so like you know very well, when an electric current is supplied, the positive charges will start to move towards the negative electrode, and obviously the negatively charged particles will start to migrate towards the positive electrode. So you know very well, DNA is a negatively charged particle and they will start to migrate towards the positive electrode, that is anode. So here in this case, for the migration of the DNA from one terminal to the other terminal, we need to provide them some sort of medium, that is supporting structure should be provided. Here we make use of Agarose gel as a supporting medium or matrix. So basically this Agarose gel is the one which is prepared with the help of Agarose, which is a polysaccharide derived from agar-agar, which is a seaweed. So basically the Agarose is commonly used in different concentration which varies from 0.2 to 2%. So the concentration of this Agarose gel, it mainly depends upon the type of DNA which you are going to get, which you are going to separate. So say for instance, if you are going to isolate or separate a DNA fragment which is of smaller molecular weight, then you have to opt for higher concentration so that the pore size will be higher. Obviously, when you are going to select if your intention is to isolate large DNA fragments, then you have to go for low concentration of Agarose. So that is why I said earlier, the concentration of Agarose plays a major role. So let us step into the procedure. So the first step is like casting of Agarose gel. So first we are going to select the Agarose. So you are going to take 2 grams of Agarose. So you are going to mix it with 100 ml of the buffer, which might be either TBE buffer or TAE buffer, and then you will be placing it in the oven. So you are going to just place it in the oven for 10 to 15 minutes. And the thing here you have to ensure is that you have to swirl the flask occasionally, and overboiling should be avoided, because once you overboil, the buffer will get evaporated. So that should be taken care. And the next thing is, once the boil starts, we have to just remove the flask from the oven and let it cool to 50 degrees. So after that, you have to add ethidium bromide, that is 1 micrograms per ml of ethidium bromide. So this ethidium bromide is nothing but it is a tracking dye. So this dye will bind with that of the DNA, and that will help to visualize the DNA under UV light. So once you add this dye, the next step will be like you have to cast the gel. So for that, we will be using this casting tray. So before you just drop the gel, molten gel into this tray, ensure that you are placing this well comb. So once you fix that well comb, you have to add the molten mass and then you have to allow it to cool. So ensure that it gets solidified. So it has to come to jelly-like consistency. So once you get the gel-like consistency, you have to remove that Agarose gel from the gel tray and you have to transfer into the electrophoresis unit, or you can say it as gel box. So when you place the gel into the gel box, ensure that this well should be closer to the negative electrode, because as I said earlier, you're going to isolate and separate the DNA, which is a negatively charged one. So you have to place this well very close to the negative electrode, so that the DNA will start to migrate towards the positive electrode. So once this is done, once it is placed, you have to just add the buffer, TAE buffer or TBE buffer into this electrophoresis box. So you have to be very careful when you're going to drop the sample. So the first well should always contain the reference ladder, or you can say it as DNA ladder. So it's also called as reference DNA, and you have to make use of the micropipette, and you have to load this reference DNA, and followed by that you have to add the DNA samples. So before adding the DNA samples into the well, you have to add a little bit of bromophenol blue. So this dye will monitor the electrophoresis. So you have to mix 1 drop of this bromophenol blue dye with that of DNA sample, and then you have to load it in the well. So the first lane will have the DNA ladder, followed by the DNA samples to be examined. So here, once it is loaded, you have to just turn on the power mode, and then the loading dye will just migrate towards the positive electrode. So you have to make sure that when you select a loading dye, the loading dye should move faster than that of the DNA sample. So nearly 5 to 10 volts is selected here. So once the dye has reached the positive electrode, you have to just turn off the power supply, because it ensures that the DNA has come and it has migrated. So once it is done, you have to just turn off the power and remove the gel from the gel tray. And since we are using ethidium bromide as a tracking dye here, please make sure that you are wearing the gloves in removing the gel. Then it is placed, the gel is placed in the UV illuminator, and once it is turned on, you can just make out the brightly colored orange bands over here, which indicates the presence of bands of DNA. So this is about the visualizing the sample. So once this is done, next what we are supposed to do is, see, we have isolated the DNA now. So you have to isolate the DNA based on the size. For this, you know very well, we are going to load the reference DNA here in the first lane, and the remaining will be the DNA. So here in this picture, let us see, this is the DNA ladder, and these are the DNA samples. So say for example, if I am going to just separate this DNA, I have to just cut this DNA along with the gel, and then you have to just compare this band with that of the reference band. So this is how you find out the length of the DNA fragment or size of the DNA fragment. So once this is done, you have to just say, my intention is to isolate or cut this DNA of this particular size, then I have to cut this along with the gel and then go for the extraction of DNA. So that procedure is called as elution. So this is a very important procedure which is done in rDNA technology. So now let us see about the applications of this Agarose gel electrophoresis. So the application, in the sense, you know very well, like this is the one which is used to estimate the size of DNA molecules, and even it is used for the analysis of PCR products, especially during fingerprinting, and even for molecular genetics diagnosis, we go for this Agarose gel electrophoresis, and even for separation of restricted genomic DNA as well as for RNA with the help of the southern analysis and northern analysis respectively. And these are the sources which I have used for the reference. Thank you.